מוסיפים אחר כך שוב, אני אסביר לי.
מה לגבי מתי?
כן, לגבי מתי.
רגע, אבל מה יקרה? את לא יכולה לעשות את זה בשני בסוף? אם בא אמרה לך?
אם בא אמרה שאי אפשר.
מתי שיעור מתחיל?
זה ב-20.
הוא עוד לא העלה את המאמר הזה שיש לי פה.
נכון, אני אגיד למה
הוא לא העלה כלום, כאילו.
אין לי מושג, אני עכשיו נכנסתי לעמוד היה את זה.
נכנסתי במקרה היום, והוא לא העלה פשוט שום דבר עדיין בכלל. גם לא תלוי בשום.
רגע, מתי המאמר? המאמר יוצא בסוף? בשבוע הבא? כן, וואו.
מה?
בסדר, 17 זה הבחירות.
נכון.
המאמר הראשון של
18.
כן, אני יודעת, אני לא בטוחה שאני מגיעה בשביל זה. זה אומר שדיברנו מזה אחרת שחשוב להגיע, נכון? שחובה להגיע.
זה 4.
זה תלוי איפה אני מופיעה, שאתה לא יודע מה זה.
נכון.
בסדר, אבל אפשר דווקא משם.
זה יום אחרי הבחירות?
לא?
כן. אין לי איך להצביע פה.
אם את נכה.
אני לא נכה. מה קטנות ראש?
אני
יחכו בתורים האלה בגללי.
לא, באמת שלא אני כי ידעתי בתשובה וצחקתי. אני לא בסתמות, באמת בסתמות, שאני יודע, אבל זה יש לי סוף.
סטודנטים יש לי סוף, פשוט שואלת, אבל זו אחת מהבחירות איפה את רשומה?
תגיע, יש לך סוף נכון.
ששאלו אותך, נט ולא כולם ספורים, אז בשנייה שלא לא, תמיד אתה שואל את השנייה?
לא, בסדר, אני לא יודע איך הילד זה, אני יודע איך הילד.
גם נסיעות וזה לא רשום, יש אופציות שלא תהיה מודברים, רק לומדת פה, ואני כן חושבת שאני שייכת
לא, מתי את רוב הדברים
יש לי צעיר אחר, אבל אני כן רוצה להשפיע עליה בבחירות המקומיות.
הוא לא סומך עליי. מה?
ועל זה הולך?
מה המצב?
יכול להיות שלא בסדר.
דוגמה.
לא מבינים מה הייתה הקריאה הזאת 20 דקות.
האלה.
כן, אני גם לא, הבנתי שהייתה איזו הודעה, אבל אני לא קיבלתי אותה. זה היה פשוט, אנחנו התקבלנו בניסויים, הם דאגו שידליזו.
האמת שאנחנו לא התקדמנו.
זה אומר ששוב,
אנחנו לא התקדמנו, אנחנו התקדמנו.
לא. לא, אנחנו צריכים לחזור לניסוי.
לא, אמרה לחזור עד 3:20.
כן? אני לא צריכה שלא נדבר בתוכנית. אני צריכה שלא נדבר בתוכנית, מותר לעשות את זה. נכון, זה די בעייתי. יש כאן גבול, כן, אפשר אחת. למשל. כן, זה ממש
אני לא אחזר למחשב.
אז איפה עשית את הסטודנטים? מבחינת עיקרון ספרדית ומדבר.
לאט לאט. גם עשיתי שם קורס, גם ראיתי שם בסיסית את כל
לא,
התקשורת
אני גם
הבסיסית,
מדבר.
תקשורת ברחוב.
אני מדבר ספרדית, אבל אני לא יודע אם נגמר, זה בכל זאת עברה אז. ואולי...
פשוט...
אולי זה משהו שפשוט מבין.
מההתחלה זה לא...
תראי אם יש אותה
כמו
ירוקות.
לא צריך את הפיצה הזאת בשביל
בצד אחד גם היה
כאילו,
אחד.
בטיר שיש בו המון חמצן, וגם נותנים את זה נורא כיף, נורא
זה
טעים.
בסדר.
בא לי מלא, באתי פיצה.
כולם מדברים ספרדית.
האמת שהייתי
יש כאן בחור שכתב בחור, ואני מאוחדת להיות פעיל וחבר בקבוצה,
נורא שונה,
אם לא משחרר אותנו, הוא לא נותן לאף אחד
כל מיני אירועים, אנדלוסיות, כל
בלי להפסיק.
תקופה
אתה לא יכול לשתוק יותר.
אז זה בהתחלה
זה לא להשתיק עוד שלך?
להתחיל לדבר
חושבת שגם היה בזה זה יכול להיות יותר
אני יודעת שאם הייתי נשארת שם עוד סמסטר, אז כבר הייתי מתחילה אותו.
בכל
תגידי, מה זה, היית באריתוס מונדוס הזה? כאילו
שעכשיו כבר אין לזה תואר ראשון, הייתי
נשמע כמו הוגוורטס.
לא צחקו עליכם שהגעתם עם כמה בוחנים כאלה הייתם? כמה בוחנים כאלה הייתם?
3.
תמר לא הגיעה עם חברה בסרטונים?
תמר אם היא הייתה מגיעה למסיבה, והיא לא הגיעה, אבל היא הייתה מגיעה עם הספסרות שבראש כזה.
לא, לא צחקו עלינו כל פעם. לא כזה צחקו עלינו.
דמות
נכון, אבל למה
טלוויזיה רוסית.
חלש?
3. הייתה תחפושת, לי והייתי שלה, ולימוד בתחפושת העסק.
טוב, עכשיו צריך לקבוע תאריך הסדנת הוקרה.
נכון.
לא אנחנו, אבל
יאיר,
24.
זה הזמן להרים את הסלוגן.
איזה סלוגן?
בגלל שאני אבא אני לא זכאי לשוקולד.
אתה מילא, הוא באמת הוא רצה להיות, הוא התכוון להכין.
הם הזיזו את
חפרו לו
התאריך.
בזה.
הוא אפילו גזר קצת.
הם פשוט שינו את התאריך ברגע
חפרו לו
האחרון.
בפייסבוק.
אמרתי, הם פחדו שאנשים לא יבואו, כי הוא לא מודיעים
זה נכון. היו יותר אנשים שיצאו. כאילו גם אצלנו היו.
אנחנו נצאים פה שעות כבר.
מסיימים, אנחנו מחכים לכם.
כן, אבל זה עדיין לא הוגן לעשות את זה ברגע האחרון.
לא, זה מטופש.
כן, הם אמרו שמטופש כמו שאתה לא יודע.
הם הודו בזה. זה לא כמו יאיר.
אבל נו גם
רציתי להגיע.
אתה לא רצית להגיע בשום שלב, לא משנה באיזה יום זה היה. הזיזו את זה ליום שלישי, במיוחד סליחה שלך ללכת ליד בגילה, זה לא מדהים.
אפשר להגיד לי יום לפני.
הוא מתוכנן. הוא לא יכול לתת כל הזמן.
אבל לא יודעים מה זה דברים דינמיים, לא, הכל לפני
מה? לא תוציא את הילדים
אני אומר
שלך.
לבית שאני מצטער. פתאום אני אומר לו, לא מצטער, זה לא יודע,
אני פסיכולוג. תביא אותו לפה. היו בזמן הילדים.
הכוונה היא ש... בדיוק. שיהיה לא טוב.
לא בגלל שאתה האחרונות.
מחכים לכולם.
רביד וטובי.
טובי לא רוצה.
רביד וטובי, כן, בדיוק עכשיו סיימו לחתוך עם הגברת קופי.
תפקיד לי, אל תצחקו עלינו, זה לא יפה.
זה לא עליהם.
נכון, למה אומרים לא מזכיר?
הוא אמר שהוא לא יזכיר.
כן, נשים לא אומרים את זה בעיה. כמה
אמרנו 30 שקלים לעומרי.
יאיר, אתה רוצה עכשיו?
בדרך כלל 200 שקל, אפשר להתחיל? שיש עוד בדרך.
יש
שניים.
בעצם מזוהים לדבר הזמן ולא נוכל ללמוד. כן, נכון? כמה
הנה.
מה זה למטה בדרך?
אין עכשיו בשלבי שנייה.
את צריכה להצביע.
את מחוץ לעסק.
כן.
את
רגע. אני בתואר ראשון. יש לי אישור מיוחד.
אוקיי.
אבל יש לך שיעור בשתיים?
יש לי שיעור, כן.
בשתיים?
אני יכולה גם ללכת אם אני יכולה. יש לי שיעור בשתיים, כן.
פה בבניין?
כן, כן.
ונכנסנו כאן משני צדדים.
אז הם בדרך, הוא אמר יש לנו עוד 2 דקות.
איזה קורס נפש.
נראה לי שעדיף לעשות את זה.
עד מתי?
לא נראה לי שיש ברירה.
עד מתי?
עד 2. ואז יהיה מתי שהוא לפעול כאילו שם זה.
נתעסק חזרה בצורה זוכרת?
לא, אני מביא לך 200. אני לא יודע שלך כסף.
אני לא יודעת אם שעת אני הולכת. אז אל תביא לי.
מזל טוב לכל הנשים פה לרגל יום אישה בינלאומי.
גם לא קיבלנו בבוקר.
טוב, אם יש לך שם 200,
הם מפסידים.
מה אתה?
תראה את זה בחזור.
לא, אני משאיר 200 בכוונה.
לא,
כמו שבא.
לא, המייל שלי של הפלטינדיאדה הוא אחר.
זה של המוביל.
כן, זה בלי נקודה באמצע, בלי נקודה או משום מה.
אני לא יודעת. אני לא
אני אמרתי שאני לא אסכים.
מישהו מאיתנו זה היום פתרון. בסדר, מישהו אחר היה יכול לקחת. הוא צודק לגמרי. גם להוציא את הכסף וגם אין רשימה, כל אחד פה מצפון הפרטי שלו. זה לא מצפון, זה זיכרון. בסדר, אז מסכימים, נו. הנה, את יצאת מזה, זהו. נעשה מדי פעם מוסכמות קבוצתית כזאת. צריך לעבור אחד אחד לבוא. אני חושבת ששילמתי, אבל... לא, זה אני שמתי לי, אני שלח בבית שלך בלכתי.
אני רוצה להגיד.
אני לא הכנתי אותה. אני מסכימה את החברה שלי. חשבתי שהיא אמרה את ממש טעימה. פה, 30 שקל הקטנה שלך.
באמת ממש טעימה.
כן.
נתי, שיית עצמי 37 כל זה עצובה, זה
תכננתי
טוב, אוקיי, אני אתחיל. Should I speak English or English?
English.
Okay, so we have three lessons, 3 meetings where I will give a lecture, and then the fourth one, which is going to be delayed due to some local organization, you will present, or 3 of you will present works, papers that will be decided around the topic that I'm going to talk about in the next 3 lessons. So it's today, Sunday, Wednesday, 12 o'clock, and next Sunday again at 12 o'clock. I believe that there is no lab anymore during this. It's no lab. Okay, so it's 12 o'clock. Okay, so I want to discuss with you some classical plus recent advances in looking at neurons as computational elements. So I want to look at the neuron. I want to introduce the neuron to you in terms of its electrical properties and some other interesting properties, and then discuss the possibility that neurons compute, that they are not just simple input-output devices, but more sophisticated nonlinear input-output devices as we think about them today. So the idea today is to think about the neuron, the element, unlike in physics where the element is very simple and the computation is done globally in the network, which is of course the case also in the nervous system. But the question is whether in the nervous system the element itself, the neuron itself, is already performing computation on its own before or as part of the collective computation. Right. So that's my mission, so to speak, to show you how we think about neurons. But first to introduce to you some of it. Some may have heard some of it because you came from different backgrounds. Some less and some more. So tell me if you feel that it's too slow or already known. There is a lot of material anyway, so we can go faster. Okay. So the plan of as I see what my role here is to do the following. I want to introduce you always like to introduce, to start with some history, so that you don't think that things have started yesterday. Just to show you what Kahal and Golgi role. So these are two opponents, the anatomical opponents, and these are two physiological opponents, or how should I say, big fighters between themselves. And I want just to introduce you this controversy, and then just brief introduction to neurons in terms of what is important in order to understand the computational aspects of neurons. Mostly the electrical properties of neurons, the cable properties of neurons, as it is called. Then introduce to you the passive cable property, passive cable theory. Did you hear about passive cable theory? Some of you know passive cable. How many heard about it? Under what? Where did you study that? Who taught you that? Who?
Your course on
No. My course in I'm asking here.
Cellular basis in the first semester.
Who gave Okay, so I'm going to go very fast about that, and just to give you some of my intuitions through working with the cable theory, because they will be relevant to understanding the computational aspect. And then I want to add some recent results that you may have heard, some of you about and some not, because we are getting more and more into the capability to record from dendrites, which was not something we could do before. So we can do direct recording from dendrites, both optically and electrically, and we start to see that most of the complexities, actually, in terms of nonlinearities, are in the dendrites rather than in the axon. So I want to introduce you the nonlinearities in dendrites, and then I want to discuss computational aspect of neurons. Okay, so that's the idea, so I just want to start with these two characters, because all that we know about, or most of what we know about neurons started then. So you know that in 1906 there was the first Nobel Prize in neuroscience, medicine, neuroscience. So Nobel Prize started in 1901, first Nobel Prize ever, and 5 years later these are the two first people who got Nobel Prize for their work on the nervous system. And you know that they were very, very big opponents in terms of thinking about the nervous system, which is very important because it's not so clear what was the controversy. Even on the podium, they fought when they got the Nobel Prize. They didn't agree with each other. You know that Ramon y Cajal, the Spaniard, believed that there are neurons, separate elements, separate from each other, and Golgi thought that it's a continuum, physical continuum, and a big network of elements touching each other. And it seems like a trivial controversy, but they understand that it's not a trivial controversy, because of course the question is what is the elementary computational unit in the brain? And if everything is connected physically to everything, it's not clear to know what is the element. We know today that neurons are mostly, not always, but mostly separate, because there are these chemical synapses between them. You know that in some systems neurons do touch each other via gap junctions, and these gap junctions are electrical connection, for example in the retina. Recently we know that inhibitory neurons in the cortex and in the hippocampus are connected via gap junctions, so there are local networks of neurons that are electrically connected in a big networks, electrical networks, rather than chemical networks. But typically in the cortex and other systems, we know that chemical synapses are separating one neuron from another. So eventually, of course, Ramon y Cajal was right in a sense that neurons do not interact directly, but there is something that is passing between them. He, of course, did not know anything about synapses, so the term synapse came much later. The anatomical basis of the connection, synapse, came much later. They didn't have EM at that time, of course, because they only used, so this is Ramon y Cajal in Spain, and this is his microscope. He used very, very, very primitive microscope relative to what we have today, but still his imagery, he was imagining that there are separate elements, and he started to draw neurons in a very beautiful way, exactly at the same time that, the same years that Van Gogh started to draw the trees in Arles, in south of France. Ramon y Cajal started to draw neurons because he was the first to use first. He was using Golgi stain. You know that Golgi, the other guy, developed a stain still used today. And this stain stains neurons sporadically here and there. So this is one neuron stain, another neuron stain, and many, many others that are not stained. And luckily, because otherwise it would be completely black. So this Golgi stain luckily stain about 1% of the neurons for some unknown reason as yet. Some neurons absorb this stain, some neurons do not. And so Golgi and Ramon y Cajal saw neurons in the most beautiful way, and Ramon y Cajal and Golgi started to stain them, to draw them, just by looking at the microscope and drawing with the other hand. So they saw it and they stained and they drew, and they got these beautiful drawings. So this is old drawings, elaborate work by Ramon y Cajal. He drew hundreds and thousands and hundreds of thousands of neurons from all regions, from the cerebellum. So these are well-known cerebellar neurons, the retina, you know, the bipolar cells and the receptors and other cell types and the cortex and so on. So he was really a big painter. He was an artist. Also Golgi. We don't know much about Golgi because his writings were not translated to English as much as the case of Golgi. So we know much more about Ramon y Cajal first. Golgi was also a genius. Now I want to go into the functional aspect of Ramon y Cajal, what he called the neuron doctrine. So the neuron doctrine is the fact that we have separate neurons, but beyond that, so neurons are independent of each other, and you can see that Ramon y Cajal drew these arrows. So these are original arrows by Ramon y Cajal, and he saw that this axon, for example, coming from the thalamus. So the thalamus is down there, and this is the cortex. So this is the skull, this is the pia, this is about 2 millimeter deep. Okay, so this is the cortical sheath, which is very thin. This is layer 2, 3, and this is layer 5 and 6, and so on. But he saw axons coming from the thalamus, and he believed that they are not touching, this axon does not touch the neuron that he sees. Of course, there are many other neurons here, as I said. This is Golgi stain, so there are many other neurons here, and in principle, this axon could touch other neurons that are not here. But anyway, this is the drawing, and you can see that this arrow shows some jumping of something, some jumping, so to speak, from activity here to activity there. But they don't touch each other. They come close to each other, but they don't touch each other. Note, very interesting and very importantly, that Ramon y Cajal already drew in his dendrites, in his pyramidal cell dendrites, you can see these spines. You learned about spines, these little appendages, about 10,000 or 8,000 per one cell. So he already saw these spines. He already saw in this primitive microscope, he saw these little spines coming out of dendrites. Also here, these little appendages, so dendrites are not smooth, but they have these little appendages coming out of them. These little branches, small branches, about 1 micrometer in length. And he thought, Ramon y Cajal was really a genius because he imagined how the nervous system works without really knowing anything about electrical currents or anything like that. It's all anatomy. Just anatomy. There is no recording, there is no electricity here. It's all a vision about how information, whatever it means, information, flows here. So it says it flows from the axon, jump to the dendrite. Then the dendrite is the receptive area. So his claim is the dendrites are receiving inputs from axons that are the output. And so that's what he called the theory of dynamic polarization. The dendrites are polarized. They are polarized by the input first, and then the input flows into the soma, and from the soma to axon of this cell, and from this axon of this cell to dendrites of another cell. So the whole concept that we are still used to thinking the dendrites are the input sides and axon is the output side is coming from this theory of dynamic polarization. It's first polarized here electrically, maybe, and then it flows to the soma, and from the soma axon, dendrites, axon, dendrites. We know it's not true. We made a mistake here. We know about backpropagating action potential, So arrows can also go backward. We should discuss it later. But at least in principle, it's true that most of the synapses impinges converge onto the dendritic tree of neurons. In many cases, most cases, we know the dendrites are receiving inputs via one directional synapse. So it releases presynaptically and it accepts postsynaptically about 10,000 inputs.
Isn't it also incorrect in the sense that it's depolarization and not polarization?
Yes. He didn't know, of course, about polarization, inhibition, and excitation, and two direction, and so forth. So polarization, dynamic polarization. Not depolarization or hyperpolarization, but just polarization. You polarize this region electrically, he thought, and then the flow of current goes this direction, which is not completely true, but we'll discuss it in a second. As I said recently, due to optical advances, we can record, for example, from this Purkinje cell. So you can stain the cell with fluorescent dyes, for example, lucifer yellow in this case, and then you can see the dendritic tree in a slice, also in vivo today. You can really patch, you can come with an electrode, you can see how big the electrode, so to speak, is relatively to the dendrite. The tip of an electrode is about 1 micrometer. So it becomes near the diameter of the dendrite. So it's a problem like you put a big nail into something that is as big as this, you can break it apart. So typically people record it from cell bodies, and that's what we knew until 20 years ago, only from this location, which is big relative to the electrode. But today we can patch at some locations. Some experts, not in Israel, but there are some big experts in the world that can patch electrodes in several dendritic locations and see the flow of information locally in the dendrite before it comes to the cell body. Michael Hauser is one of them. There are others, few others, because this is a very elaborate thing to patch a dendrite, and at the same time to patch an axon, and maybe even to patch the axon, which you don't see here, but there is an axon coming out here. Just to show you axons, because I know that the tendency sometimes is to think that dendrites are very elaborate and axon is just like a wire, כמו חוט חשמל, like electrical wire, going through, but it's not. So you can see the green thing here is a dendrite. If you know how to converge your eyes, you can see three-dimensional. People can do it sometimes. If you know how to converge, you will see the axon coming to you, because this is a little diverged, a little angled, one relative to the other. But anyway, the dendrites are the green thing. This is a pyramidal cell in the visual cortex of the cat by Kevin Martin. This is the dendrite, and you can see the local axon. Of course, the axon can go elsewhere, but locally you can see the axon of this particular cell coming out and branching and branching, and you can see how extended is the axonal branching near the cell, near the region of the cell, and of course also elsewhere. But you can see also that this axon has these yellow boutons. This is the release site. This is the presynaptic release site in the axon. So you can see how many synapses are being exerted by this axon. So this axon, when there will be an action potential going here, there is a probability of release from here, a probability of release from here, from here, from here, from here, from here. So this should give you a perspective on axon. Is a very sophisticated, very elaborate axonal tree with many, many release sites. Many, many release sites. About every 4 microns you have one release site, another release site, another release site. So one action potential that goes through may release here and there and here and there and here and there, talk to the network, so to speak. This is only the local axon, as I said. In many cases, especially if you are layer 5 pyramidal cell or layer 2-3, your axon may go to the other, both to other regions, also back into the thalamus, depending on which neuron it is. So I just wanted you to think about axon as a multi-site release and not a wire that goes through a tree. But it covers many, many trees, like branching, branching, branching. This could be, the postsynaptic tree can be in one tree. The postsynaptic tree of this bouton could be another tree, or maybe the same tree. We don't know from this.
Are there known neurons that have axons that connect to their own dendrite?
Yeah, in most cases there are autosynapses. So in most cases that we know, the axon is also talking to yourself, but with a very few synapses relative to the total synapse you receive. So if you receive, let's say, 10,000 synapses, 10 synapses may be your own. In some cases it could be interesting in some kind of a loop, but so typically they don't need necessarily to avoid your own dendrite, but the contribution is small relative to the rest. But yes, this synapse, you may make synapse on your own dendrite in many cases. So as I said, there are several experts today, and I will show you that because of this capability to record from at the same neuron, this is work from Matthew Larcombe, who is probably the best in the world to be able to patch the same dendrite and also the soma and sometimes also the axon, and just to understand how information flows from the input side, so to speak, to the output side, and also discovering through this, the backpropagating action potentials. You all heard about the backpropagating action potential, which is a new signal that we did not know about until 15 years ago or so. The fact that the action potential that starts here also goes backward into the dendrite. We should discuss it because that's a very important plastic signal in the nervous system. Anyway, so people can do this today, very rarely, but people can do this today, and you can see what is happening here. For example, the discovery of calcium spikes in dendrites came out of this, and things of that sort. We should discuss it in a second. As I said, I want to look at the dendrite, at the neuron, with all its synaptic inputs and complicated dendritic structure, and with all its complicated axonal structure, asking whether this unit, on its own, could perform specific computational functions. That's really where I'm heading in this short introduction in the next three talks. So I want to also highlight something that I see, at least in my field, as a huge, amazing, fantastic advance, maybe the most interesting paper that I read in the last few years. This is a paper in Nature by Arthur Konoth. Arthur Konoth is working in Munich, in the University of Munich, and he is really a big developer of refined technology, and he's pushing two-photon microscope. Two-photon microscope is now a very popular tool, expensive tool, but very popular tool. There is no lab that is serious without two-photon microscope. Electrophysiological lab like Yossi Aron now and Avi Mizrahi and others, Eli Nelson, are using the two-photon microscope that we have here. But they typically record with two-photon microscope, they use calcium imaging to record from the cell body. So whenever there is a spike in the cell body, or in the axon, just in the beginning or outside of the cell body, in the beginning of the axon, there is also flow of calcium inside the cell. Although it's a sodium spike, some elements of it are also calcium that are going from outside to inside, and you can image the excess of calcium into the cell body. So this is what is called calcium imaging using two-photon microscope. Usually people focus on the cell body, like Adi Mizrahi, Eli Nelson, Yossi Aron, they focus on the cell body to see which cells are firing, so to speak. Because if this cell becomes fluorescent, it means that calcium went into here, so there was a spike here, or spike here, or spike there. So you can use the two-photon microscope today, as you know, hopefully, recording from a network of neurons that are active. Some people reach, like Raphael used to, up to 1,000 neurons. You can image very fast, and then you assemble all this data, and you know which network is now responding to this sound, or to whisker movement, or whatever system you work on. But Arthur Konoth pushed the system, so to speak, in terms of resolution, to the level of imaging individual synapses. Okay, so what he did really in this paper, as I mentioned, he pushed the two-photon microscope resolution to be able to image the calcium in the dendrite of the postsynaptic receiving cell. So there is a cell there, here is the cell, there are the dendrites, and you can image locally here, very, very fine focus two-photon microscope, postsynaptically, so you record postsynaptically. Today you can even record from a single spine. So it's from a single spine. So he imaged a single spine. I hope you understand that this is a very high resolution. So this is your dendrite, and this is the spine coming out of the dendrite. So he's imaging calcium in the spine head. And the synapse is sitting here, yes? So he can see the activity, so to speak, of a single synapse when the synapse is active and the transmitter is released and there are NMDA receptors and whatever, and there is calcium flow inside, you can image calcium at the level of a single synapse. Is it clear? So it's a much, much higher resolution because all this is much smaller than 1 micrometer square, much smaller than 1 micrometer square membrane area. And the cell body is much bigger in terms of membrane area or volume. But this is a very small volume here that you have to image the calcium concentration from here, and that's what he succeeded to do. Why is it so important? Because what you really want to know eventually, let's say in the visual system in this case, is what is the input, not only the output, what is the input to this particular cell? Because of course the question is in terms of computation. So you have a cell, this is let's say layer 2-3 cell, this is the cell body here, this is the dendritic tree, this dendritic tree in the visual system V1 of the mouse visual system, this is layer 2-3 cell, and this is one synapse, this is another synapse, this is a third synapse, impinging on this dendrite. Eventually, this cell is directional selective cell. Let's say that this cell is selective, has a receptive field around, let's say, 45 degrees. So the output of this cell is easy to record. You can impale with an electrode here and put different angles. Okay, you can move your grid in this direction, or in this direction, or in this direction, or in this direction, as we do. So the mouse is sitting and looking at the screen, and you can shift bars in this direction, and this direction, in this direction. You can record from this cell intracellularly, as people can do today, in vivo. And you can say that this receptive field of this cell is around, let's say, 45 degrees. But you want also to understand how the input is converging to the output. You want to understand what is the input here. What is the sensitivity of this synapse to what angle is the sensitivity of this particular synapse, or of this particular synapse, or of this particular synapse. This is a big jump, because you really, it's the first time, not the first time, I'll show another example. It's one of the first times where people, autocolons, succeeded to both record the output of a cell and at the same time record, in this case, from about 20 inputs to this cell at the same time. And so the question is, how does the cell takes the input and converge it to a particular output with this sensitivity? What is the computation being done in the dendrites? Is it a majority winning? So let's say that 90% of these inputs are sensitive to 45 degrees and some are sensitive to 50 degrees. Is that how the neuron computes? Or maybe all these synapses are sensitive to the same direction, they are directional, selective to the same output. What is the computation between the input to the output?
Did you record it from 20?
Sorry?
You recorded from 20?
In this case, about 20, yes.
But we assume that there's more than 20, no?
10,000.
So
10,000, yes. Well, we have to progress a little bit more. But I'm saying, you're right, of course. You sample very, very briefly. I'll show you what is the result of this. I'm saying that it was a big jump, because before we didn't even have a signature of the input. We didn't know what the input is saying. We only knew what is the output of this cell. So I characterize a cell as being cell with this directional or orientation selectivity of 45 degrees or 50 degrees, but I didn't know what is the input to this cell. So this is the first time that we really recorded the input to this cell. It's not the first time, because it was done before by Axel Borst, also in Munich, on the fly visual system. I'll show you in a second. But I'm just trying to tell you that this was a big jump, technological jump, to be able to at the same time record the output and also the input to the dendrites of the same cell. This is what I wanted to say.
And I don't fully understand why you call it a single cell computation, because the reason that this cell fires more for a single orientation is because of the inputs it gets.
question.
other orientation.
That's the question.
Whether it's
That's
the
exactly
input
the
or the
question.
computation of the cell itself?
That's exactly the question. Do I respond, I'm now the output, do I respond to 45 degrees because I get more 45 degrees as an input? Do I do some average of many inputs and get some average? Or is there some nonlinear boosting, for example, that takes the few that are 45 degrees, let's say, and boosts them and makes them? That's exactly the question that I'm trying to understand. What is the conversion? What is the computation between the input? What is the rule of conversion between particular input to a particular output? What is the rule? We don't know that. We just don't know that.
So
We never knew that before, and we still don't, as I will show you, because of this, exactly this issue, that there are only few, we only have a sample of very few inputs out of many other inputs, and so we don't really know. But I'll show you in a second what we do know.
So, sorry.
Yeah.
So, in that sense, we're not really sure yet that there is such a thing as a single cell computation?
No, there is a single cell computation because eventually this cell becomes responsive to 45 degrees, let's say.
Yeah, but
And we don't know how it computes these 45 degrees. What is the algorithm that it uses in order to take whatever the input is and eventually sharpen its output to become sensitive to 45 rather than, let's say, to all direction. We don't know.
But if the input you get are stronger 45 degrees, like absolutely stronger than other stimuli, then it's a network computation. The inputs are what determining the orientation of the cell, not the cell itself.
That's the question, exactly.
So if you're not sure,
So I'm saying that there is some local mechanism in the neuron that select. Maybe, I'm saying maybe, this neuron selects in some particular way a subset of the inputs and use them to generate an output. It could be. Or that it's completely determined by the input. There is no local computation, but it is, determined by the input. For example, majority win all. You just count 1, 2, 3, 4, 5, 6, 45 degrees. Only 2, 30 degrees, so the majority wins, and that's the computation. So you just take and you compute, you count. I call it computation as well, but it's a trivial computation in some sense. And we don't know that that's what happens. We just don't know. I just know to record from the output that that's the output of the cell. But somewhere there must be computation, either at the network level or at the single cell level or both. That's exactly what we are trying to understand. We claim that there must be local nonlinearities that sharpens the properties of the neuron itself, take what it sees from the environment, so to speak, from the network, and sharpen it, add the computation on top of it, and make it more specific, let's say. That's the claim, but I have to show you that that's what happens. And we don't know. But I'm saying that this kind of directions, or this kind of technology, and starts to enable us to ask questions. I will show you what are the results for this limited but new. But as I said, I think it's the most, in terms of understanding neurons, it's one of the most advanced things that happened in the last few years. Is this capability to record directional selectivity, which is shown here, by the way, I didn't mention it. But when I show you this, I mean that this synapse, so to speak, or autocolon means that this synapse has a preferred orientation in this direction. And let's say this synapse, which is this one, has preferred orientation to this direction. So you can see already from this picture, which is not trivial on its own, that it's what we call salt and pepper organization. Salt and pepper, meaning that the neuron in the visual system of the mouse receive all orientation, many orientations on the dendrites. You could think, one possibility was that all the synapses have the same direction selectivity as the output. For example, we believe, but nobody did it yet, that in the cat visual system, Jubel and Wieser, you heard about Jubel and Wieser recorded from the cat V1, and there is a whole column, so to speak, a column, functional column, not anatomical column, but a functional column, where the orientation selectivity of all the cells, or most of the cells in this column, are the same. Let's say 45 degrees, of all the cells. So if they talk to each other locally, then you would expect that all the synapses for a given cell are also directional selective for the same orientation in the column of the cat. So there it's not salt and pepper. Here it's salt and pepper. We know that the visual system, you heard the term salt and pepper before? So we know already before this experiment, doing calcium imaging of cell bodies, of cell body response. So if you recall from the cell body calcium imaging of two-photon microscope, let's say Clay Reed from Harvard, now in the Allen Institute in Seattle. If you recall from this cell and this cell and this cell and this cell and this cell, image the cell body selectivity. In the cat, as I said, most of these cells will have, if they are nearby, in the same functional column, most of these cells will have the same orientation selectivity. If you are in the mouse V1, and you recall from this cell and this cell and this cell, they will have completely different orientation selectivity. There is no organization of a columnar functionality, as in the cat. So this is already called salt and pepper, meaning that this could be a salt and this could be a pepper in terms of orientation selectivity. And this could be a salt, but many types of salt because it's all the orientation. So this could be a red cell, let's say 45 degrees. This could be a blue cell, let's say 30 degrees orientation selectivity. Green cell, salt and pepper. That's the term salt and pepper. So in some sense it's not surprising that also the synapses are salt and pepper, because the synapses are coming from nearby cells that they themselves have different orientation selectivity than you. But still, somebody had to show it, and he was the first to show it, as I show you here, which is a really elaborate work and very, very particular. They went also to do the same thing, by the way, for the mouse, not for the cat, with Ellie Nelkin. They did it on the auditory cortex. They found salt and pepper organization for frequencies. They went also to do it in the whisker system with Gerd Sackman to show the salt and pepper organization, that each cell receives input from different whiskers. I'm not going to go into it, I'm just summarizing what they found. You can see, you could think of many, many types. You could think, for example, that one dendrite, it could be that one dendrite will receive orientation, let's say, of this angle. That the mapping of a given angle will be per dendrite. So this dendrite receives, I don't know what, 45 degrees. This dendrite receives, let's say, 90 degrees. This dendrite receives synapses with orientation selectivity of 135 degrees and so on. But this is not what they found. This is what they found. They found that nearby synapses on the same branch could code for different orientation. Okay, so this is an interesting finding. Anatomically, there is a full, so to speak, mix of orientation, selectivity of the input to the dendrites, complete mix. So two nearby synapses could come from a different input type cells. And this will tell you 45 degrees, this will tell you 50 degrees, this will tell you 75 degrees, and so on. And eventually you, as an output, becomes very sensitive to a particular angle. And as I said, we don't know yet because we don't have enough about the input, but we know that it's some kind of a mix. It receives many, many, many directions. Yossi Arom will tell you, for example, that tells him that this cell can very rapidly switch, maybe after plasticity, from becoming sensitive to 45 to becoming sensitive to 60, because it already has the input available. It's only a matter of making this one and this one and this one stronger, let's say, then you become sensitive to this direction. Or you make this one and this one and this one stronger, then you become sensitive to this direction. That's one possibility. The other possibility is, as Arthur Connor's recording from this cell for many, many hours sometimes, it's very, very stable. It's always 45 degrees, keeping 45 degrees. Of course, he didn't do yet plasticity things to show that this particular cell can become sensitive to another direction. But anyway, it has the machinery. You can see it has the machinery. So this is a very, very nice work. Really, I really like this work a lot.
So where do you find these neurons?
So this is in the visual cortex V1 of the rat, of the mouse. This is V1
But you said
of the mouse.
But you don't see them in functional columns of the cat, you said?
No.
And
We don't. Nobody did this. Nobody did calcium imaging at the synaptic level in the cat. I'm just saying that thinking, first of all, that in the cat, when you record from cell bodies, calcium imaging at cell bodies, you see that all the cell bodies respond. There is no salt and pepper organization in the cat.
Right. So I mean,
So there is a functional, like Wieser said, that there is all these cells in terms of output. But probably also in terms of input, if I would record from the cat, I would assume, nobody did it, that all the synapses will have the same orientation selectivity as the output of this cell.
right. So
Nobody did it.
Yeah. So I'm trying to understand what the purpose of a salt and pepper system would be. In other
What's
words,
the purpose?
Yeah.
That's first of all, he sees. So there is another solution to the cat or to us, rather than having this column, that all the columns, so to speak, respond to the same orientation. There is another way to solve it. It could be still that upstream, there is columnar organization, and these two cells from the salt and pepper, let's say this one and this one, that are responsible for this orientation, are connected together to this one. So the column is mixed, so to speak. You don't see it at this level of V1, but maybe you see it at this level, because after all, the mouse has to be able to respond to this angle and not to this angle.
Right.
But it's mixed at this level.
Yeah, but like the input that comes through the retina
And thalamus. Retina, thalamus, V1.
Yeah. So already in the retina, right? Isn't there already some sort of orientation information there?
Yes.
Because of
some ganglion cells in the retina, which are the output of the retina to the thalamus, some ganglion cells are already directional selective, orientation selective. Some of them.
Right. So for early visual process, like I understand later downstream if you have like conflicting information, so you might want to put it all together and have some sort of function which takes different inputs and gives an output based on some function. But we were talking about basic sensory input. Why does it make sense to have all these information about different orientations coming into the cell and then deciding which one it is? And I'm trying to figure out where in the processing it makes sense to do that.
I'm giving you an anatomical fact.
Okay.
The salt and pepper. I can give you also the fact that although the mouse is not as good in terms of vision as the cat, which could be also related to this organization or something else, I have no idea. But I'm saying that eventually the mouse also can distinguish between different orientations, so somehow it can decipher out of this mixing balagan, so to speak, a soup of orientations within one region. Somehow upstream he is able to look, so to speak, downstream and tell me that this is now 45 degrees, but we have no idea how. I'm just saying that this is a functional anatomical finding that deserves an understanding about how is it being done. By the way, if you already asked me these questions, I should tell you that there is now even more advanced work by students of Conrad and others that succeeds to record from two cells with the same orientation selectivity in the mouse. So they have to find this 45 degrees and let's say this 45 degrees and asking what is the connectivity, lateral connectivity between these two cells. They are starting to show that locally, if you have two cells that are sensitive to this orientation, they tend to be more strongly connected to each other than this cell to this cell. Okay, so there is local connectivity between cells with the same orientation preference within this salt and pepper balagan. So this is another new feature that is emerging from being able to record and reconstruct the connections. Huge work, yes, not only to find the two of them, but to record the strength of synapses between the two of them as compared to somebody with a different orientation. All this is upcoming. This is the front of trying to understand visual processing in the mouse visual system using this technology. But as I said, it didn't come yet to the cat or to other animals as yet, this technology of imaging orientation selectivity, or imaging the receptive field of individual synapses. That's the thing, of the input itself. That's a new technology, 3 years old or something. Already 5 because we are 2023. Another fellow who does, and we are working with him is, sorry.
Can I ask a question about the previous
Yes.
thing?
Mm-hmm.
Are these
Yes. So the calcium imaging, you know, with two photon you can decide in advance. Of course, there are many limitations. It has to be on the same focal plane, although today there are 3D two-photon microscope that can change very fast the focus. But let's say the 2D two-photon microscope, like Adil is doing, it has to be on the focal plane. And you can decide that I'm going here, I'm going here, I'm going here, I'm going here very fast. So I'm jumping here, tick, tack, 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 almost at the same time, not at the same time, but at a frequency of, let's say, 100 hertz. So every 10 milliseconds I jump from one to the other, and I can record simultaneously, or almost simultaneously, the receptive field of all these synapses. At this plane, if I need to go deeper, so to speak, then I have to change the focus Z-plane, and I go to another plane, and then I record. But it doesn't really matter if it's simultaneously or not, assuming that it's not changing from time to time, but just you want to focus on different dendrites, on this synapse, maybe on a deeper synapse, and then reconstruct, project the dendrite on 2D and see if this could be deeper than this one, and this could be deeper than that one. But this is the organization that they found, not this. This is the organization that they found.
Yeah, the reason I'm asking about time is I'm thinking that maybe there are temporal patterns that important.
With two-photon imaging, calcium, as you know, there is a huge problem of time in terms of spiking, so to speak, because the timing here is it's not on the spiking level. Until calcium goes in, collated by whatever the calcium collator is there, the fluorescence is not at the millisecond range. So in terms of millisecond resolution, the two-photon microscope is not doing good work. Electrode is. But we cannot implant electrode here. This is too much. So this is a limitation. So you're right. It could be that there is some organization in time. We should discuss this, and this could do specific computation. The timing is correct. That's one way to do it, for example, in terms of computation, because if this and this comes together, and this and this comes later, it could be that just the timing determines what would be the output. But this we don't know from this type of experiments because of the calcium, which is a very slow process. Hundreds of milliseconds.
Hundreds of milliseconds?
Many tens of milliseconds. They are trying to push it faster because of the dyes that will respond faster to the calcium entry and fluorescent fast, but it's... For example, Adi and Eli are quite certain that they can, using the calcium signal from the cell body, as I said, they can distinguish whether there was one or two spikes. Not exactly the timing of the spike, but whether there was calcium elevation via one spike or calcium, but not exactly when the spike happened. So there is a jitter of several tenths of milliseconds, maybe more, about the timing of the spike. So that's a problem of imaging, of calcium imaging. There are new imaging of voltage-dependent dyes. There are new dyes that are voltage-dependent. And these dyes respond to voltage, not to calcium concentration, but directly to voltage. These are faster dyes, but there is a whole issue of signal to noise. Because the calcium imaging is a volume response. You have the concentration is volume. The voltage-dependent dye is a membrane, so it's a surface, and there is an issue of but people developed it. And it will become more and more available. Then it will be wonderful, because then you really can record the voltage at each location very fast too. This will give us a new perspective on neurons, but we are not there. Okay, slower than I thought, but it's good. I just wanted to tell you that there is another wonderful work by Axel Bors in Munich, Max Planck Center. You know that we have a Max Planck Center here, part of this Max Planck Center there. And Axel Bors was actually the first, really, to be able to record from the fly visual system. You know, the fly is extremely good with vision in terms of succeeding to maneuver in 3D and chasing another fly and not going into the wall and things like that. The fly is a very good, behaviorally very good in terms of vision. And Axel Bors is really the leader in the field of understanding the different levels, processes from the receptors and the different layers, until he can record from this type of cells, calcium imaging, from individual inputs, regions. So he can record calcium from this part of the dendrite, from this part of the dendrite, and he knows that the input from the world, so to speak, from the world, which means after several layers, is this region, and this region, and you can look at the receptive field of, let's say, branch number 1. So you can see that branch number 1 responds strongly in terms of calcium when there is something moving in this direction in the world. The branch number 2, which is this branch, likes this direction. The branch number, I don't know what, 9, which is this branch, likes this direction. So you can see that this one branch in the fly visual system receives input from different angles in the world, so on different regions of the dendrite, which is a little similar to what I said before, but here it's more kind of systematic. There is something systematic about the change of sensitivity as a function of location. So this is like looking at the world, being sensitive to this direction of motion. Here it's sensitive to this direction of motion. And eventually the output here in the axon is again sensitive to a particular direction. Out of all these possibilities. So now I personally have a student now who modeled this, trying to understand what type of computation. Is it just linear vectorial summation? You just add vectors. You take this vector plus this vector plus this vector plus this vector and compute the output. Is it just linear vector? Or is there some kind of amplification? For example, could that number 9 is closer to the output than number 2 because it has to go through a bigger cable, a longer cable? Does that mean that the weight of number 9 on the output is more than the weight of number 2 on the output? So then you start to talk about computation. So it's not just linear sum, but it depends on the location of the synapse, for example. And maybe there is some nonlinear boosting in this region and so forth. So that's what we are trying to understand, how this output cell, which is very important for the fly, has 10 like this, and if you destroy some of them, you won't be able to see vertical lines or horizontal lines. It's going to lose it. So we're trying to understand the computation because we know the input, we know the output, and we can try now, and we have a model of the cell, which we should talk about in the next lesson, table model of the cell. We can try to understand better how the computation is performed from the input sensitivity, let's say to this direction or to this direction, to the output sensitivity to some other direction. Okay, so I want just to tell you a few words. I don't know why this is so low resolution. Anyway, terrible. I don't know, maybe I should do something else. Anyway, it's much lower than my computer, so you can't read it very well, but I'll put it, of course, I'll put it on a link so you can download the lecture. So you don't really try to read it. I just want to highlight what is important in my mind. So this will be the morphological column. And in this morphological column, I try to summarize what is important in terms of dendrites, in terms of morphology, just for you to remember numbers. For example, the length of a dendrite, typical length of a dendrite, is over 1 millimeter. Okay, it's not 1 centimeter, and it's not 100 micrometers. A length of a dendrite, when I say length, I mean air length, okay, cavaviri, from the soma to the tip. It's about 1 millimeter. Could be half, depending on the type of dendrite. But this is the length. So dendrites are local. Dendrites are very local. Axons could be very long. Axons could be centimeters or sometimes even meters. An axon that starts here in my motor cortex can go through my spinal cord until the toe. One axon. So axon could be very long, and dendrites are always very local. And these are the numbers here in terms of how long the dendrites here, what is the surface area of dendrite, because you know that most of your brain are dendrites in terms of surface area. In terms of length, you have more length of axons. You know that you have 4 kilometers of wire in 1 cubic millimeter. 4 kilometers of wire in 1 cubic millimeter. So these are the typical numbers. How many spines you have, what is the size of a spine, what is the volume of a spine, just to give you general numbers. Now about physiology. So in order to discuss cable, which we shall discuss later, in order to discuss cable, maybe you have to ask Nizar why it's so low resolution. I need to know 3 basic parameters to discuss the cable properties, the electrical properties of a neuron, the passive electrical properties of neuron. I have to know the specific membrane resistivity Rm of ohm multiplied by centimeter square. So somewhere here, membrane resistivity. So this is Rm, you see here Rm. I give you the range, the physiological range that people measured, the Rm. You can think about this Rm as the resistance of a square, centimeter square of membrane. So suppose I had a centimeter square of membrane, which I don't, of course, I have much less than centimeter square. But if I had a centimeter square of membrane, so 1 centimeter or it's 1 centimeter, this is what is called the specific membrane resistivity, specific because it is per unit area. This is the units, and these are the numbers. So it can go up to 100,000 ohm centimeter square. This is a very, very, very resistive membrane, or it can be very low resistance, like 5 kilo ohm centimeter square, 5,000 ohm centimeter square. Why this is so important? Because Rm multiplied by Cm, which is the specific capacitance of this membrane. So there is a membrane, there is Rm and there is Cm, which is the capacitance of a square centimeter square, and this is a rather universal number. It's always near 1 microfarad per centimeter square. So this is multiplied by centimeter square and divided by centimeter square. And if you multiply Rm by Cm, Rm multiplied by Cm, this will give you, so the area disappears because it's one over the other, and you get units of time. This is the time constant of the membrane. The time constant of the membrane, of course, is the key, the most important element in terms of integrating inputs. So if I have a neuron with a very fast time constant, it cannot sum up inputs because the inputs decay very fast. If I have a slow time constant, let's say 100 milliseconds or 50 milliseconds or 20 milliseconds, I can sum up inputs because the decay time is slow enough, so the next input, if it comes near enough on time, I can sum up. So this Rm multiplied by Cm, which gives me the tau membrane, is the key parameter for neurons. It gives you the clock time of the neuron, so to speak, in terms of summing inputs. So this is why when we speak about the cable properties of neurons, I need to know Rm and Cm in order to know tau.
You mean like local RM and CM, because it can vary along the...
I'm assuming now, which is an assumption, which is not a correct assumption, that it's the same membrane all over the dendrites per unit area. The same membrane per unit area as a first assumption, and so the time constant is uniform. The membrane time constant, because it's the same membrane. Sorry?
This is how we think of it.
That's how we think of this. We know that it's not always the case. In some cases, the membrane is more shunted, so Rm is lower. In some places, but as a first assumption, when we should speak about cable theory, we should assume it's the uniform cable. In terms, it's the same membrane wrapped into a cable, even if it's thinner or larger, it's the same membrane. So it's the same membrane area. By the way, of course, the input resistance, which is another parameter of the cable, the input resistance, which is in the case of an isopotential sphere, is Rm divided by the area. Of course, also depends on Rm. So the input resistance, whenever you have a small area, a very, very small area, this becomes a very big input resistance. So, for example, the input resistance of the spine, which is a very, very small area, becomes very big. The input resistance of the cell body, which is big, the input resistance of the cell body is not as big as in the spine. So these are the numbers here, for example. You see the input resistance can go, if you record from a cell body, just to give you numbers, which is of course very important in terms of how much voltage is being generated as a function of the current being injected to the cell. So the cell body, typical input resistance is 100 megaohm. If I record from a cell in the cortex, it will be 50 megaohm, 100 megaohm at the cell body. 100 megaohm means that if I inject nanoampere, nano is 10 to the power of minus 9. If I inject nanoampere into the cell body, I will get 100 millivolts. 100 mega ohm, 100 to the power of 6 ohm, 100 mega ohm is 10 to the power of 8, yes, multiplied by 10 to the power of minus 9 will give you 100 milli, 0.1 volt, 0.1 volt. So you see how much, very little current, very small current, will give you significant voltage in the cell body. But if you're in the spine, where you have a synapse, and the spine input resistance is giga ohm, because it's so small, then you need much, much less current in order to generate big EPSP, big local voltage. So that's why these numbers are of course important, because you want to know how much current you need to generate, what kind of voltage and so forth. I'm not going to go into the details, of course, but this is just to give you some kind of insight about what are we talking about in terms of numbers, mega ohms in the cell body, giga ohms in dendrite, in the input resistance, time constant in the cortex of 10, 20 milliseconds. The time constant of a typical cortical cell will be 10 or 20 milliseconds and so forth. So this is what is written here basically. There is another parameter that I should mention. This is Ri. So this is the third key parameter in cable theory, Ri, which is the axial resistivity. So it's not a membrane property, but rather the cytoplasm property, the medium inside. Okay, so this is Ri. So if you have a cable, of course, and there is a longitudinal current flow, of course you can either go outside of the membrane through the capacitance, charge capacitance, or go outside through Rm channels, or you can go longitudinally. And the question is how resistive is the axial resistivity, which is Ri, and this is going, as you will see there, something on the order of 100 ohm centimeters. Anyway, these are the three key parameters, Cm, Rm, Ri, if you want to talk about passive cable properties. There is also active properties, excitable properties, we should discuss a lot, because as I said, and that's amazing that we don't understand exactly why, most of the interesting ion channels, the calcium, voltage-dependent calcium, the sodium-dependent calcium, sodium-dependent, voltage-dependent, and sodium and potassium-dependent, and fluoride, and all these four types of ions are flowing in the membrane, so to speak, calcium, potassium, sodium, and fluoride. But they have many types of channels. We know, for example, for potassium, just for potassium, we have something on the order of 50 types of channels. And when I say type of channel, I mean that the sensitivity of the ion channel to voltage, or the kinetics of the channel in terms of opening and closing, is different between one channel and another. So why is there such a design of so many types of channels for only 4 types of ions to go out, in, and out? Why do we need so many kinetics and so many voltage sensitivities for these ion channels, especially in dendrites? So most of these sophisticated ion channels, H channel, T channel, P channel, H channel, and all these names, and more are becoming known, are mostly embedded in the dendrites. And you remember that these channels are being generated in the cell body, the protein, they are being sent to the dendrites, or also to the axon, because they need to generate a spike in the axon, so you need sodium dependent and so forth, and also calcium in the synapse and so forth. But most of them are in the dendrites. Many tens of different ion channels are distributed in the dendrites. Why? Why do you need all these nonlinearities with different kinetics to put in this input zone? Why? That's an open question. But it's a fact. So I'm just giving you some of these, typical channels, calcium and potassium and sodium. But excitable properties are becoming more and more clear, as you recall from dendrites, and you can patch a membrane and really can look at the channels. You can see more and more interesting type of channels expressed in dendrites. We know that certain types of diseases, for example, like epileptic seizures, may start because you have too much H-type channels, which is mixed type of channels, potassium and sodium type in the dendrites. So these nonlinearities could be also functionally important, to do certain computation, but also can, if they go out of balance, you can generate certain diseases, like like uh, like seizures and other types. So this is something that is being worked on now with molecular biology. There is a whole project called Channelpedia to develop an encyclopedia, so to speak, channelpedia of the channels, because these are the source of current flow in neurons. You need these channels to open and close in order to get spikes, to get EPSPs, to get IPSPs, to get electrical current. You need these channels, but apparently nature devised a lot of different types of channels for different purposes we don't really know about. It's not very clear. How does it affect the dynamics of the network, the different type of channels? We know it doesn't affect, but we don't know much. Or of course there are the synaptology, which type of channel of synapses are impinging on dendrites. You know that typically we have two types of synapses. This is the most clear distinction, excitatory and inhibitory synapses. This is already known from Sherrington, another Nobel laureate from 1930s, British physiologist, who discovered the synapse, who gave the name to the term synapse. Sherrington, but he was the one also to discuss different types of synapses, and his whole Nobel Prize lecture is about the balance between excitation and inhibition. So this is the two types of the big synapses types in the brain, but of course inside these types there are many subtypes of excitatory synapses and inhibitory synapses. For example, we know that there is these two types for excitatory synapses in the cortex. We know that there are two types of channels of receptors, the AMPA receptors and the NMDA receptor. Both are sensitive to glutamate. It's the same transmitter, but once, so to speak, received by AMPA, which is a fast receptor, once maybe being received by NMDA receptor, which is a very slow receptor, until it catches, so to speak, the transmitter. It opens and then it closes very short, very slowly, on the order of 100 milliseconds. So already at the level of synapse, you have at least two time constants. One very fast, AMPA, and one millisecond range, and one very slow, hundreds of milliseconds, 50, 100 millisecond range. This is AMPA and NMDA, and surprisingly, also in the inhibitory synapses, where GABA is the main transmitter, you have the GABA A receptor, which is a receptor mostly open to chloride and is very fast, relatively fast. There is the GABA B receptor, which is a receptor mostly to potassium, which is very slow. So that's something good to remember, that at the input already level, you have in terms of synapses, of time course, you have slow and fast excitatory, you have slow and fast inhibitory synapses or receptors. So this is what I really wanted to highlight in this, without too many details, just for you to get some kind of general understanding about what we have. Let me start with the controversy and then we shall break. So these are the two big competitors. So I told you about Ramon y Cajal and Golgi as the anatomists who fought and didn't agree. You know, Ramon y Cajal, Golgi was a romantic person. He was Italian. He liked opera. So he didn't like, really, you're right. I can't think of a nervous system like any other living tissue which he knew is built from cells. So the term cell is from the 18th century, the term cell. The tissues, living tissues, are built from individual cells. Muscles or skin are built from cells. But he refused to accept that the brain is also built from cells. It's another tissue. It cannot be that the opera and emotions are coming from cells. It must be something unified with something romantic here. And these two fellows fought on something else. So Wilfried Roll is the one who developed cable theory for neurons. Cable theory was developed for undersea cables by the physicist Thomson, which we'll discuss in a second, but he took the cable equation, the physics equations, and he used them to understand current flow in neurons, dendrites, nerve cells. And Eccles, who got Nobel Prize for his work on the spinal cord, wonderful work, he refused to accept dendrites as something important functionally. He said, of course, there are dendrites. We knew that there are dendrites from the time of the dendrites in terms of anatomy, but functionally, he felt that they are not important. And what he says is really the following. So we know that this is a dendritic, this is a drawing of Ramon y Cajal. This is the most well-known drawing of any neuron ever. If you can see it in any book, in any lecture, in anywhere, this is Ramon y Cajal drawing of an axon and a dendrite of Purkinje's cell. This is a wonderful cell, beautiful cell in the cerebellum. It's an inhibitory cell, by the way. The output of this cell is inhibitory. It's a wonderful cell. And this is how cells looks like, or maybe this is how cells look like. We know that they look like this from the time of Ramon y Cajal and Golgi. People drew them. But in terms of how we think about the neuron, this is the way we think about neuron until today. So this drawing from books is not just because you want to reduce the complexity, which you want. You don't want to draw, each time you draw a neuron, you put all this complexity, especially if you draw a network. It will be a bush, it will be a jungle. So you make it simple. But you put all your, look, you put the synapses on the cell body, but you know that the synapses are here. You put the synapses on the cell body, and that's how you think about the neuron. And that's how Eccles thought about the neuron. Now, when I'm saying thinking about neuron, modeling in your brain the neuron, thinking about neuron in terms of the modeling how the neuron behaves, this has a very significant, one physiological significant meaning is the following, that if you have something like that, let's say that it's isopotential. Because it's so small that everything is isopotential. There is no voltage difference between this point to this point. So this is called isopotential. So if I draw a neuron like this, I think of the cell body as isopotential. That all the membrane has the same potential at any given point of time. This means that the attenuation of voltage is exponential. Because if you write, this I hope you know, that if you write the equation for an isopotential RC membrane, you get one time constant out of it, which is the time constant of voltage decay, which is an exponential voltage decay with a time constant tau. So if my model in my brain, so to speak, or Eccles' brain, the models in Eccles' brain, which was the model, is an isopotential cell, and let's say that I inject the current here, or maybe a current comes from dendrites here, but this is isopotential, then the voltage response here, and the attenuation of voltage at the end of the current, is governed by a single exponent, e to the power of minus t divided by tau m. One exponent. Just because you have one compartment, there is one time constant governing both the development and attenuation of voltage. And because you think like this, all your experimental results of Eccles, the experimental results that Eccles recorded from, so Eccles recorded EPSPs from the cell body of a motor neuron. So the motor neuron is very complicated. Motor neuron is the spinal cord. It's very complicated with many, many dendrites. And he was one of the first to record intracellularly from here in the cell body of a motor neuron. And he saw an EPSP that looks like this, and it decays. So this is an EPSP, maybe coming from the activation of this synapse, or from injection current of your own current. He looked at the attenuation, and he forced on the attenuation one exponent. He said the attenuation is governed by one exponent. Why? Because the model in my brain is of an isopotential cell. That's my model. Okay? So he forced, and he made several mistakes, because he forced a model that is an isopotential model on something that is absolutely not isopotential. Now, why it is not isopotential? And that's what Roll, who was his postdoc, talking to each other for many years, because Eccles refused to accept a paper that Roll wrote in '59. He was a physicist, Roll, in Manhattan Project. He came to neuroscience, like Heinz Polinsky, realized that neuroscience is interesting. He came to neuroscience with a very strong physical background, Wilfried Roll, and he was very, very simply said, look, I have a cell body, and I have dendrites. Most of the membrane is in the dendrites. You saw the huge dendritic tree and the very small soma, relatively. In terms of membrane area, the soma is maybe 5%, maybe 2% of the total membrane area of the dendritic tree. I'm not talking about the axon, which could be huge. But in terms of membrane area, the soma is 5%. So how can it be that my current, let's say I inject current here, of course my current will flow elsewhere, to the place where you have more conductance. It will not go through the membrane of my soma, most of it. So it cannot be isopotential. It means that if I inject current here, there will be another voltage here, because there is a current flow and there is attenuation of voltage from the place where I inject the current to another location. So that's how we started to develop the cable theory, because he says that dendrites are distributed electrically. They are distributed, they are not isopotential. There is a synapse here, it generates local voltage. There is a soma there, it sees another voltage, completely another voltage. There is huge difference, and we shall show you next time what is the expected difference between the local voltage to the somatic voltage. So how can I relate to it as an isopotential element when it's not? Just by looking at it. Then of course he did the computation and he built the cable theory and so on. So this was his really first kind of example why most of the current, doesn't matter by the way if the current